Good morning everyone. Myself Dr. Devangi Dwivedi, Senior Lecturer in the Department of Oral Pathology and Microbiology, BBD COTS, BBD University. Today I am going to tell you about life cycle of ameloblast. According to their function, the lifespan of the cells of the inner enamel epithelium can be divided into 6 stages: morphogenic, organizing, formative, maturative, protective, and the last is the desmolytic stage. First, let's take up morphogenic stage. Before the ameloblasts are fully differentiated and produce enamel, they interact with the adjacent mesenchymal cells, determining the shape of the dentinoenamel junction and the crown. During this morphogenic stage, the cells are short and columnar with large oval nuclei that almost fill the cell body. The Golgi apparatus and the centrioles are located in the proximal end of the cell, whereas the mitochondria are evenly dispersed throughout the cytoplasm. During ameloblast differentiation, terminal bars appear concomitantly with the migration of the mitochondria to the basal region of the cell. The terminal bars represent points of close contact between cells. The inner enamel epithelium is separated from the connective tissue of the dental papilla by a delicate basal lamina. The adjacent pulpal layer is a cell-free, narrow, light zone containing fine argyrophil fibers and the cytoplasmic processes of the superficial cells of the pulp. Next is the organizing stage. In the organizing stage of development, the inner enamel epithelium interacts with the adjacent connective tissue cells which differentiate into odontoblasts. This stage is characterized by a change in the appearance of the cells of the inner enamel epithelium. They become longer and the nucleus-free zones at the distal ends of the cells become almost as long as the proximal parts containing the nuclei. In preparation for this development, a reversal of the functional polarity of the cells takes place by the migration of the centrioles and Golgi regions from the proximal ends of the cells into their distal ends. Spatial staining methods reveal the presence of fine acidophil granules in the proximal part of the cell. Electron microscopic studies have shown that these granules are actually the mitochondria which have become concentrated in this part of the cell. At the same time, the clear cell-free zone between the inner enamel epithelium and the dental papilla disappears, probably because of elongation of the epithelial cells toward the papilla. Thus, the epithelial cells come into close contact with the connective tissue cells of the pulp, which differentiate into odontoblasts. Recently, it has been shown that the pre-ameloblasts secrete proteins similar to those of enamel matrix. These proteins appear to be phagocytosed by developing odontoblasts. It is suggested that these proteins may play a role in epithelial mesenchymal interaction. During the terminal phase of the organizing stage, the formation of the dentin by the odontoblast begins. The first appearance of dentin seems to be a critical phase in the life cycle of the inner enamel epithelium. As long as it is in contact with the connective tissue of the dental papilla, it receives nutrient material from the blood vessels of this tissue. When dentin forms, however, it cuts off the ameloblasts from their original source of nourishment, and from then on they are supplied by the capillaries that surround and may even penetrate the outer enamel epithelium. This reversal of nutritional source is characterized by proliferation of capillaries of the dental sac and by reduction and gradual disappearance of the stellate reticulum. Thus, the distance between the capillaries and the stratum intermedium and the ameloblast layer is shortened. Experiments with vital stains demonstrate this reversal of the nutritional stream. Coming to the formative stage, the ameloblasts enter their formative stage after the first layer of dentin has been formed. The presence of dentin seems to be necessary for the beginning of enamel matrix formation, just as it was necessary for the epithelial cells to come into close contact with the connective tissue of the pulp during differentiation of the odontoblasts and the beginning of dentin formation. This mutual interaction between one group of cells and another is one of the fundamental laws of organogenesis and histodifferentiation. During formation of the enamel matrix, the ameloblasts retain approximately the same length and arrangement. Changes in the organization and number of cytoplasmic organelles and inclusions are related to the initiation of secretion of enamel matrix. The earliest apparent change is the development of blunt cell processes on the ameloblast surfaces which penetrate the basal lamina and enter the predentine. Coming to the maturative stage, enamel maturation, that is full mineralization, occurs after most of the thickness of the enamel matrix has been formed in the occlusal or incisal area. In the cervical parts of the crown, enamel matrix formation is still progressing at this time. During enamel maturation, the ameloblasts are slightly reduced in length and are closely attached to enamel matrix. The cells of the stratum intermedium lose their cuboidal shape and regular arrangement and assume a spindle shape. It is certain that ameloblasts also play a part in the maturation of the enamel. During maturation, ameloblasts display microvilli at their distal extremities and cytoplasmic vacuoles containing material resembling enamel matrix are present. These structures indicate an absorptive function of these cells. Coming to the protective stage, when the enamel has completely developed and has fully calcified, the ameloblast cease to be arranged in a well-defined layer and can no longer be differentiated from the cells of the stratum intermedium and outer enamel epithelium. These cells layers then form a stratified epithelial covering of the enamel. This is called as reduced enamel epithelium. The function of the reduced enamel epithelium is that of protecting the mature enamel by separating it from the connective tissue until the tooth erupts. If connective tissue comes in contact with the enamel, anomalies may develop. Under such conditions, the enamel may be either resorbed or covered by a layer of cementum. During this phase of the life cycle of ameloblast, the epithelial enamel organ may retract from the cervical edge of the enamel. The adjacent mesenchymal cells may then deposit a fibrillar cementum on the enamel surface. Last is the desmolytic stage. The reduced enamel epithelium proliferates and seems to induce atrophy of the connective tissue, separating it from the oral epithelium so that fusion of the two epithelia can occur. It is probable that the epithelial cells elaborate enzymes that are able to destroy connective tissue fibers by desmolysis. Premature degeneration of the reduced enamel epithelium may prevent the eruption of a tooth. Here is the diagram showing the different stages in life cycle of ameloblast. That was all. Thank you.